Hey everybody, welcome to chapter 17. We are going to be talking about immunological disorders today, particularly the hypersensitivities, and then both autoimmune and immunosuppressive disorders. We'll also talk a little bit about treatments. So let's go ahead and get started. Hypersensitivities are these are a reaction when the immune system is overreacting. The immune system is has been sensitized to something, and then this in what we call a primary exposure, and then the second time that someone is exposed, they have this kind of overwhelming, over exaggerated type of immune response. So the immune system is just kind of going overboard. Now there are 4 types of hypersensitivities. Type 1 is what's referred to as an immediate or anaphylactic reaction. Now when we think of anaphylaxis, we think of allergies to things like bee stings or say penicillin. Those are injected allergens or systemic anaphylaxis. There are other kind of forms or less severe forms of anaphylaxis. This includes hay fever, asthma, even some skin reactions. So it's really actually called an immediate IgE mediated hypersensitivity, also more commonly known as an anaphylactic reaction or type 1 hypersensitivity. Type 2 is a cytotoxic, so you can tell here that cells are going to be destroyed at some point in time. So type 2 is called a cytotoxic hypersensitivity. Type 3 is immune complex mediated. So just from the name of it, you know that now we're going to have things like combinations of complement and antibodies and immune cells all kind of clumped together to create large complexes. And then type 4 are the delayed type cell mediated responses. These are things like some skin reactions, and they are more T cell related than anything else. So let's take a look at each of these. Oh, sorry, moved up a little ahead. So in type 1 hypersensitivities, the immediate IgEs, we have our first exposure. And in first exposure, the antigen that we refer to in hypersensitivities as an allergen, such as pollen, pet dander, dust, mites, those sorts of things. The allergen, the person will first be exposed, and the allergen will activate the adaptive immune response. Here we have an antigen presenting cell, so this could be a dendritic cell or a macrophage. It is going to present antigen to the T cell. The T cell is going to say, all right, not a problem. Present to the B cell. The B cell says, okay, makes plasma cells. And what happens here is instead of the plasma cell pumping out IgG antibodies, or IgMs at this point, this time the plasma cell pumps out IgE antibodies. Now normally we would see IgGs against something like a pollen or an allergen, but instead the plasma cell, when it class switches, ends up class switching to IgE antibodies, which then bind to the surface of mast cells and basophils, mast cells being kind of in the mucous membranes, basophils usually being in tissue. The second time you get exposed, these IgE antibodies that are sitting on the surface of these mast cells and basophils will get cross-link bound. That's what's happening here. So the two antibodies will get bound. This causes the rapid degranulation of these mast cells or basophils. Now, mast cells and basophils are granulocytes, and those granules that are already within the cell contain histamines. These are what we call mediators. So histamines will be released immediately. And histamines do things like vasodilation. They cause smooth muscle contraction, excess mucus production, all of the things that you would associate with a typical allergy response. So if you have kind of a hay fever type response, sometimes they call this allergic rhinitis, you would inhale an allergen, a pollen, or cat dander, dog dander, and this would be the pollen. And the first time you're exposed, usually very early in life, your B cells, for some reason, instead of creating IgGs against it, for this particular response, it created IgEs. So these IgEs on those mast cells and basophils found in the mucous membranes and in the tissue in the upper respiratory system will degranulate immediately. And this is why if you have allergies like hay fever or pet dander, you know immediately. You walk into a room, you're allergic to cats, you get this kind of tingling sensation that occurs as soon as you inhale. And that tingling sensation is the immediate release, the IgE mediated release of those histamines. Once histamines are released, the cell produces secondary mediators called leukotrienes and prostaglandins. And both leukotrienes and prostaglandins further enhance the effects of the histamine. So if we have a lot of basophils and mast cells being released, being degranulated, the more you have being degranulated, the higher the concentration of those mediators. Greater the mediators, greater the allergic reaction. So we can have people who are really super sensitive and they just pump out tons of mediators and they have really potent reactions. We have people who are, you can have people who are slightly sensitive, so they get, they may not have as many IgE antibodies, or they just don't produce as many mediators. So these mast cells get activated in secondary exposures, as do the basophils. One thing to remember is in IgE-mediated type 1 hypersensitivities, this allergic reaction is only going to occur after primary exposure. The person has to be sensitized first. So the first time you're exposed, you don't have an allergic reaction. You don't even know. But the second time you're exposed, and every exposure thereafter, because all you're doing is producing more IgE antibodies, you're going to have those types of allergic reactions.
Immediate response. Type 1 hypersensitivity reaction. A hypersensitivity reaction is when the body responds to an otherwise harmless substance which is normally ignored. In this case, the body recognizes the substance and triggers an inflammatory response. The usually harmless substance is called an allergen. The inflammatory response is colloquially referred to as an allergic reaction. An immediate allergic reaction is a type 1 hypersensitivity reaction and requires pre-sensitization. During sensitization, the body's immune system initially comes into contact with the allergen. In tissue, the allergen binds to receptors present on B cells. Part of the antigen is transferred to an MHC molecule and presented on the B cell surface. When the receptor of a T helper cell binds to the complex, the T helper cell is activated and releases cytokines. These cytokines activate the B cell and initiate its maturation into a plasma cell. The plasma cell produces and releases IgE antibodies. These antibodies have two variable regions which enable binding to the specific antigen, here, the allergen. With its constant region, the IgE antibodies dock onto a surface receptor on mast cells, which are now able to respond when re-exposed to the same allergen. The individual is now sensitized to the allergen. Upon re-exposure, the allergen binds to the IgE receptors, cross-linking adjacent receptors. This leads to mast cell activation, inducing the release of vesicles filled with inflammatory mediators. This process is termed degranulation. The three most important inflammatory mediators released are histamine, prostaglandin D2, and heparin. There are various routes in how an individual can be exposed, for example, through inhalation or skin contact. Depending on the affected tissue, the released mediators can cause bronchoconstriction, leading to respiratory distress, vasodilation, reducing blood pressure and leading to edema formation, and local inflammation with heat, redness, and itching. One of the special features of type 1 hypersensitivity reactions is that once sensitized, the response to the allergen is immediate and can occur within seconds or minutes of exposure. Other types of hypersensitivity reactions are slower and can develop after hours to days.
So you can see these type 1 hypersensitivities, the response is quite immediate. So you brush up against something and you get within a few minutes. Usually the delivery method, like skin reactions, take longer than a systemic reaction. And there are localized anaphylactic reactions such as upper respiratory or asthma or skin. And then there are systemic anaphylactic reactions, which are things like your bee sting allergies and like penicillins, other haptens. There we go. Make sure I got the right. Okay, so in type 1 hypersensitivity, here we have our localized reactions. So we have hives, hay fever, and asthma. These are not the only three, but these are three very common ones. Hives are also known as urticaria, and they are distinctive of what's called a wheal and flare. That's this kind of round, deep redness, and it's like a welt, and it kind of raises up. That's due to that inflammatory reaction that occurs with this type of contact anaphylaxis. It's very itchy, turns red, there's a lot of heat involved. It's very commonly found in food and contact allergies. It's treated either topically with antihistamines or cortisones, or systemically through ingestion of antihistamines. I will tell you, food allergies in particular, if somebody has a food allergy, they're going to have hives. Hives are a key indicator that the reaction they are having to the food is an allergy and not an intolerance. People who are lactose intolerant are not allergic to lactose. They just can't digest lactose. People who are allergic to foods will break out in these deep hives. They even will oftentimes go into some kind of respiratory distress as well. Hay fever is probably the most common allergy among the human race. This is a response to some type of inhaled allergen. Again, the reaction is within a couple of minutes. Of course, your typical allergies are runny nose, sneezing, tingling in the upper airway, excess mucus production. The stronger the reaction, the more of this you're going to see. This can lead people with really severe hay fever can actually develop respiratory distress and go into respiratory arrest, simply because as they inhale that allergen, it starts moving down into the lower respiratory system, and you see bronchial constriction, excess fluid production, blocking of the alveoli, smooth muscle contraction surrounding the lungs, as well as any smooth muscle associated with the diaphragm. So it can actually lead to more severe types of respiratory distress, such as asthma. So hay fever and asthma can go hand in hand depending on the allergy. I have a brother who is severely allergic to cats, and he through an inhaled allergen can almost become anaphylactic because his allergy is so strong. So he has a really strong asthma reaction. So in asthma, we go from an upper respiratory tract, such as allergic rhinitis, to a lower respiratory tract type of reaction. Here, the mediators are, again, smooth muscle contraction, increase in mucus production. It has to be treated usually immediately with inhalers. The inhalers are antihistamines and epinephrine. And the epinephrine will cause vasoconstriction to slow down the edema and prevent the excess fluid, as well as some smooth muscle relaxant, so that it will relax the smooth muscle so the person can actually expand the lungs to get some breath. Now, some people do not respond so well with inhalers, and they may be on long-term treatments. And so these are called bronchodilators, and they're on long-time bronchodilators. So this would be a medication they would take on a daily basis. In systemic anaphylaxis, this is a pretty rare reaction, but it is extremely deadly. And we've all heard of or may know someone who's allergic to things like a bee sting or hornets, wasps, any of the flying toxin-producing Hymenoptera, they're called. This can cause a condition known as anaphylactic shock. Now think about everything we just learned about basophils and mast cells. Well, in this case, mast cells are in tissue, but basophils are found in circulation. So if the allergen gets injected into the bloodstream, like a bee sting or a penicillin shot, then we're going to have this basophil degranulation systemically. So everything that happens locally, think allergic rhinitis or a skin reaction, all of those inflammatory signals and mediator release now gets released into the entire bloodstream, to the entire circulatory system. This causes systemic vasodilation, which is why the person just kind of blows up and has such excess edema, smooth muscle contraction throughout the body, fluid loss, massive inflammatory reaction and response, and then this ultimately, because of the systemic vasodilation, there's an extreme drop in blood pressure, and that's why it's called shock. Shock is an extreme drop in blood pressure. This will can lead to death, and a person can die from anaphylactic shock in 3 to 5 minutes. So this is why people who are highly allergic to bee stings or penicillin, those sorts of things, they either they'll oftentimes wear bracelets, right? They're medical bracelets, as well as carry EpiPens. And that is one of the reasons a few years ago when the cost of EpiPens went up by about 350%. They went from about $200 or $250, I think it was, somewhere in that range, to near $1,000. Those EpiPens, that was a big controversy. It even went in front of Congress with that particular pharmaceutical company, simply because people die without those EpiPens, and they were not able to afford them. So it became a big issue. Food allergies are another type of systemic anaphylaxis, and food allergy reactions are slower than the systemic, than say a bee sting or penicillin shot, but they can be rapid as well, but just not as rapid as the injected allergens. So the person takes it in, and usually they know immediately. They'll feel it in their mouth, in the back of the throat. They'll even feel their throat starting to close up, usually within a minute or two. But the actual death part of it wouldn't come for they usually have about 20 to 30 minutes. This is enough time to get to the hospital. However, these people also will carry EpiPens or some type of emergency treatment that will buy them enough time to get to a hospital to get more thorough treatment. Hives will always be present in a true food allergy. That's a key indicator, and they pop up pretty quickly. Throat swelling, tongue swelling, airway blockage can occur very easily. One problem with severe food allergies is the throat and tongue get so swollen that if the ambulance is called, they can't intubate, meaning put a tube down the throat, because it's swollen shut. So they have to cut underneath it's down here at the bottom of the neck, and they do a mainline ventilation. Type 1 hypersensitivities, there's a couple of different treatments. The best and most effective, of course, is avoidance, right? Try to avoid, if you're allergic to a food, don't eat that food. Make sure also that if you're severely allergic, that you're eating foods that are not processed in the same plant as tree nuts or ground nuts. Make sure that on packages, a lot of manufacturers now, they say, hey, this is, doesn't contain peanuts, but it's manufactured in a plant that uses peanuts in something else, because the protein in the peanut that people tend to be allergic to can stick around and be on the machinery. So just because the food doesn't contain peanuts directly doesn't mean that it hasn't come into contact with it. There are people who are so severely allergic to peanuts they can't even touch anything that has been processed with peanuts. Desensitization, this is a fairly new technique they've been using for about the past 15 years. The idea is to introduce the allergen to the body in really, really small, incredibly tiny doses, and then incrementally increase it. So here's a peanut allergy desensitization treatment, and so they start out with 1 milligram, and then about 2 or 3 weeks later you take a 2 milligram. And so they're slowly reintroducing the body to this allergen with the hope that the body will develop some tolerance against it. We're trying to induce the body to produce IgG antigens as opposed to IgEs. And if we start to produce IgGs, then the IgGs, the idea is that we get enough IgGs that if that person is exposed to that peanut again, the IgGs are produced more quickly. They don't have to sit on the surface of basophils and mast cells. They're floating in circulation, and they will neutralize that antigen before it can bind to a mast cell or basophil. And the idea behind desensitization is to prevent the allergic reaction, or to at least minimize it, particularly for people who are extremely allergic. Does it work? Sometimes it does, sometimes it doesn't. It is going to be very patient specific, allergen specific, treatment specific, but that's the idea, is to slowly reintroduce the body, almost trying to recreate a primary exposure. Asthma can be treated with what are called RUMABs. RUMABs are recombinant humanized monoclonal antibodies. Now, we haven't gone over monoclonal antibodies yet, but these are antibodies, monoclonal antibodies, or MABs as they're known, are antibodies that are produced in the laboratory. So they're man-made antibodies. And what they've done is genetically engineered IgG antibodies that can bind to that constant region, that stem region of IgEs. So if we have a basophil, get my pen working here. It doesn't seem to want to work. Not sure what's going on here. So remember, antibodies are bound to mast cells and basophils with their constant region bound to the actual basophil. What they're trying to do here is create IgGs that will bind to any IgEs that get produced. And if they do, then those IgEs will not be able to bind to the mast cell or basophil, so we can keep them off of those granulocytes. There are some reactions, some medications out there, treatments that are targeting cytokines. So it's a similar idea, producing antibodies that will attach to those mediators, some of those mediators that get produced. So even if degranulation does occur, the mediators produced will be blocked and will be bound and neutralized by antibodies present. These types of treatments for asthma are only used as a last resort because they're incredibly expensive, they're difficult to come by, and they're not always successful. So antihistamines are the first choice. Corticosteroids are usually layered on top of that, and then long-term bronchodilators. Now, chromones are a very new type of treatment. Chromones are an antibody type treatment that are being designed, or actually it's a medication that's designed to block the degranulation process. And the idea is to bind this drug somewhere on, it's a, what's the term? It is a non-competitive inhibitor, to go back to Unit 2 there. It's a non-competitive inhibitor that will bind to another region of those antibodies on mast cells and basophils, causing the antigen binding site of those antibodies to change. If those change, they can no longer bind the antigen, and we will not get degranulation occurring. The antibodies will be non-functional. Type 2 hypersensitivities are the cytotoxic response. Here we start seeing antibodies binding to host cells. But instead of binding to antigens, well, they're binding to antigens, but instead of binding to something that's like the cell is infected, they start binding to self-antigens. Unfortunately, by binding to self-antigens, we get a second layer of response here, which is activation of complement through the classical pathway. So these are IgG antibodies that are binding to cells, and then of course, complement recognizes this, and so we get activation of complement. But instead of this activation of complement being against a bacteria cell, it's actually against our own cell. Now, this will cause two different things to happen. The first is it activates the antibody-dependent cellular cytotoxic response using natural killer cells. Remember, NKCs recognize antibodies bound to our own cells. But it can also cause cell death. And in cell death, this is because the membrane attack complex forms. If we have activation of complement, eventually C5b or C5a is going to activate with 6, 7, 8, and 9, and we get that membrane attack complex. And it will react against our own cells the exact same way it reacts against gram-negative cells by putting, kind of poking holes in the cell membrane, and cell contents leak out. So we get complement activation and apoptosis. Two very common cytotoxic hypersensitivities are, of course, transfusion reactions. You give somebody the wrong blood type, you're going to kill them. We have our own antibodies already present. These are one of the few antibodies we develop at birth. And we have natural antibodies against the donor red blood cells. So the donor red blood cells will be bound by those antibodies. Natural killer cells or ADCC reaction will be instigated, as well as the membrane attack complex. The problem here is that this is in the circulatory system, so we get systemic clotting, and these clots and these clumps of dead cells and cell parts will start to clog up the kidney. They'll block the glomeruli. They can also get into small vessels of the heart and cause venous damage in the heart, blood vessel blockage, clots, stroke. All of that can happen, and happen very quickly. It's actually a very rapid response because the antibodies are already in the bloodstream. You are transfusing a large amount of antigen directly into the circulatory system that already contains a high number of antibodies against it. So this reaction will be very quick. Hemolytic disease of the newborn is when you have a mother who is Rh negative. Remember, our blood types are A positive or A negative. The positive negative refers to the rhesus factor or Rh factor. So a mother who's Rh negative, if the father is Rh positive, he could pass the gene for that Rh positive to the child. So if the infant is Rh positive but the mother is Rh negative, then the mother will not necessarily have antibodies against the Rh positive in her first pregnancy. But during pregnancy, there are small tears in the placenta, and the mother does get exposed, whether it is a vaginal birth or a C-section. Either way, the mother is going to be exposed to the actual Rh of the child. There's going to be mixing of blood at some point in time, usually during delivery. This means that the mother now is going to produce antibodies against that Rh factor. And if she has a second child who is also Rh positive, then that child will be at high risk. So more often than not, a mother is treated with RhoGAM. This is not an issue anymore. If when you become pregnant, they will test your blood type to see if you are Rh negative. If you are, they're going to give you an injection of RhoGAM. It is an anti-Rh antibody treatment, and it's used to block the production of Rh antibodies.
Antibody-dependent cell-mediated cytotoxicity, type 2 hypersensitivity reaction. During a hypersensitivity reaction, the body can also respond to its own components that are usually ignored. In a type 2 reaction, this response is caused by the presence of certain antibodies. Under normal conditions, antibodies are produced during an adaptive immune response to fight off pathogens. In type 2 hypersensitivity, the antigen is located on the cell surface or is part of the connective tissue. This leads to an antibody-mediated immune response to the body's own cells. The antibodies initiate a cascade of events that can lead to cell lysis, phagocytosis, or apoptosis. Therefore, a type 2 hypersensitivity reaction is also called an antibody-dependent cell-mediated cytotoxic response. Let's take a more detailed look at the mechanisms involved. Antibody binding to the antigen primarily activates the complement system. This system comprises specific plasma proteins that act as a cascade, mediating further activation of immune cells and heightening the intensity of the response. The complement cascade is triggered by a complement protein that binds to the antigen-antibody complex on the cell. This leads to the activation of further complement proteins through cleavage of small fragments. These fragments are chemotactic, meaning they attract immune cells such as neutrophils, which bind to the antibody via Fc receptors present on the surface. These Fc receptors can detect and bind to the Fc portion of an antibody. After binding, neutrophils generate aggressive substances such as radical oxygen species. These substances cause cellular damage, inflammation, and eventually cell death. In addition, activated complement proteins can bind to the target cell surface, marking it for a process called opsonization. These marked cells can be recognized by phagocytes, notably macrophages, which also use Fc receptors to bind to the antibody. After binding, macrophages start to engulf the target cell and break it down. Furthermore, activation of the complement system also leads to the formation of a membrane attack complex, in short, MAC. This complex can insert into the plasma membrane, resulting in an influx of extracellular fluid. As a result, the cell swells and eventually lyses. Bound IgG antibodies can also be recognized by natural killer cells, often referred to as NK cells. These lymphocytes also possess Fc receptors on their surface, which bind to the antibody, leading to natural killer cell activation. The natural killer cell releases perforin, a protein similar to the membrane attack complex that inserts into the plasma membrane and causes target cell lysis. Natural killer cells also release granzymes, a group of proteins that initiate apoptosis. In some diseases caused by type 2 hypersensitivity reactions, antibodies may be directed towards cell surface receptors, thereby disrupting normal cell function. There are two cases that can be distinguished. First, the antibody blocks the receptor and prevents signals from being transmitted. Second, the antibody binds to a receptor, activating it in the process, and acts as a transmitter. Both can have severe physiological consequences. As we've just seen, type 2 hypersensitivity reactions usually cause cell death. This can either be complement-mediated or complement-independent. However, for most type 2 hypersensitivity reactions, multiple mechanisms contribute to the autoimmune reaction to varying degrees. It's important to note here that cell death via lysis most enhances inflammation through the release of cell components.
So complement-mediated, one of the reasons I really like that particular video for you is because it really pulls together the combination of what we've learned in the last two chapters, right? We have all this like complement-mediated stuff, but we also have that cytotoxic response. So we have those natural killer cells, and we have other neutrophils coming in and enhanced phagocytosis and such. So we have this like really great combination in that video. It really shows you how what you've learned is applicable just simply in type 2 hypersensitivities. Now, type 3 is what we call immune complex mediated. Here, antibodies will bind to an antigen that is soluble. This is similar to the reaction we looked at in the titer, where we had precipitation. We had that immunoprecipitation reaction. When antibodies bind to a soluble antigen, this will cause reaction of phagocytic cells, and they'll react by binding with their Fc receptors, that constant region of the antibodies. And when this complex gets too large, it results in an immune complex, and these immune complexes can start to build up and start causing issues in the circulatory system. They can lodge in blood vessels. They stick to kidney, to the glomeruli. Remember, the kidney is filtrating all of this. Also, they can get trapped and stuck in capillaries. The immune complex here, this is not cell destruction. Cytotoxic was complement destroying cells. Here, complement complement gets activated because of these excess antibodies and these complexes, and then we start to see inflammation and tissue damage. That complex inflammatory response is a really potent response. So if I have an immune complex, it's just going to keep activating. Once I start activating C1, it just follows that cascade, and we start to see all of those inflammatory reactions.
Immune complex mediated hypersensitivity. Type 3 hypersensitivity reactions. A type 3 hypersensitivity reaction is caused by immune complexes, which consist of antibodies bound to antigens. Antigens and the resulting immune complexes float freely in the blood. As a result, these immune complexes are more precisely termed soluble immune complexes. The antigens involved are either external, representing foreign antigens, or internal, also termed self-antigens. Matching IgM, or usually IgG antibodies, bind to the antigens and form complexes of various sizes. Generally, these immune complexes are effectively removed from the blood, especially by macrophages, which engulf and digest them. However, smaller immune complexes are more likely to remain in the blood circulation. The circulating immune complexes tend to deposit on the blood vessel wall, especially in small vessels. Once immune complexes are formed, they rapidly initiate the complement system. This family of plasma proteins acts as a cascade that mediates further activation of immune cells. Subsequently, a series of reactions are triggered, with the most decisive involving neutrophils for type 3 hypersensitivity reactions. Neutrophils are attracted by activated chemotactic complement proteins. Using the Fc receptors on their surface, neutrophils bind to the Fc portion of antibodies in the immune complex. This binding leads to the release of destructive substances that cause cell damage in the vicinity of the deposited immune complex. Damage to endothelial cells as well as to the basement membrane of the blood vessel wall increases its permeability, resulting in leakage into the surrounding tissue. This provokes further inflammation and subsequent tissue damage. Two distinct features help to discriminate a type 3 hypersensitivity reaction from a type 2 hypersensitivity reaction. First, the components initiating the type 3 hypersensitivity reaction are initially soluble before depositing on the surface of the vessel wall. Therefore, tissues with small blood vessels and thin vessel walls are most affected by type 3 hypersensitivity reactions. Examples include the kidney, which filters blood, as well as joints, which filter plasma to produce synovial fluid. In contrast, the antibodies in type 2 hypersensitivity reactions are directed against cellular or matrix structures and therefore have fixed targets. Second, activation of the complement system is more rapid and much stronger in a type 3 hypersensitivity reaction. Serum levels of complement proteins can therefore be used to monitor the course of the type 3 hypersensitivity reaction.
All right, so that was a nice comparison of type 2 to type 3 because sometimes they do get sometimes you can confuse them a little bit, but the solubility is really the big one, where we've got these immune complexes within kind of floating around in the bloodstream, and they're binding to a soluble type of antigen, whereas antibodies in a type 2 are binding directly to a cell surface. There we go. So in type 3 hypersensitivity, these immune complex mediated responses, one of my photos is not showing there. First is one is called an Arthus reaction. The Arthus reaction is a localized immune complex reaction. This oftentimes is associated with vaccine reactions. When somebody has a localized vaccine reaction, they get a lot of swelling and pain. It's usually very painful. And so this would be at the site of the reaction. Serum sickness is a systemic immune complex reaction. Clot dissolving streptokinase is oftentimes used to treat it. Also, another form of this is the horse serum antibodies for snakebite treatments. They'll take rattlesnake toxin and inject very small amounts of it into a horse. The horse will develop antibodies against it. They then take blood samples from the horse, extract the snakebite antibodies from it, and that becomes the antitoxin. That's the antitoxin that's given for snake bites. This is an incredibly expensive, laborious, very intense process to produce that, which is why snakebite antivenom is difficult to come by. It's usually regulated by the CDC, although some areas are going to have more of it. Like, we're not going to have a lot of rattlesnake antitoxin here in South Florida, but out in the Midwest, in Arizona and the mountains, in Colorado, Nevada, Dakotas, that sort of thing, where there's a high rattlesnake population, they're of course going to keep it on hand there. These non-human antibody treatments, these are what we refer to as passive immune therapy. And so in passive immune therapy, this is passive immunity. We're going to give you some outside immunity, whether it's non-human or human, but in passive immunity, we're going to give you antibodies produced by something else. And so it's passive. We're not activating the immune system in that sense. Type 4 hypersensitivities are what we call delayed type cell mediated. These are a lot of times skin reactions, delayed skin reactions. The actual reaction itself is usually anywhere from 2 to 3, even up to 5 days after exposure, which makes it really hard to figure out what it is that somebody is allergic to. This is why when you go to get allergy tested, they'll oftentimes they'll put lots of those little, they'll draw little numbers, and they'll, give the little pinpricks, and you have kind of this big giant back tattoo and arm tattoo where they're testing you against all kinds of different possible allergens, and what they're looking for are usually these delayed 4 hypersensitivities. This will require an initial exposure. In the initial exposure, there's no reaction. So in allergy testing, they're looking to see if you're looking for secondary reactions, not primary. This is another reason that allergy testing is kind of difficult, because maybe you might become allergic to something and that was your first exposure. That can happen as well. But they give such tiny small amounts that is not usually going to happen. Type 4 hypersensitivity involves T cells. This one is all about T cells. And so T cells become effector cells after their first exposure. So effectors, these are kind of like your memory T cells. And what they're going to do is they're going to destroy infected cells. They're looking for virus infected or cells that are giving off some kind of distress signal. The problem in delayed 4 is that they start responding to that allergen, and so chronic infections, people with chronic infections can oftentimes get tissue damage in their own tissue, not from the toxin, but because of this delayed T cell response. That can happen as well. One of the most common type 4 hypersensitivities, believe it or not, is poison ivy. Poison ivy, most people have a really strong reaction to poison ivy, and it's because they have a type 4 hypersensitivity to it.
Harmful effects produced by the mechanisms of cell-mediated immunity are referred to as delayed hypersensitivity. Delayed hypersensitivity is mediated by Th1 cells, also called Td cells, and is responsible for contact dermatitis, tissue damage in a variety of infectious diseases, and rejection of tissue grafts. A contact hypersensitivity, such as poison oak dermatitis, is initiated by contact with the antigen, which is also called the allergen. During contact, a small molecule from the plant, called a hapten, binds to a carrier protein in the host. The carrier protein with bound hapten is ingested by a macrophage, and the hapten peptide is then presented on the surface of the macrophage on a class II MHC. Th1 cells with T-cell receptors capable of recognizing the hapten peptide antigen interact with the presented antigen, become activated, and increase in numbers. When the host comes in contact with the allergen a second time, the Th1 cells react with the hapten peptide being presented on the macrophage and release cytokines, resulting in attraction of more macrophages, followed by inflammation and skin lesions. Characteristic skin lesions appear after 24 hours, reaching their peak at 48 to 72 hours after the second exposure to the plant.
So you can see that there's a delayed response there, and in type 4 delayed cell mediated, that's usually inflammatory signals. It's the inflammation that ends up causing all of the tissue damage. Get this to move here. So here's just a little bit of a close-up on that poison ivy or poison oak type response. And we have our sensitization steps, so primary exposure, antigen, we get phagocytosis of it with antigen presentation, T helper cells become these memory cells. But in our secondary response, these memory cells also we get some cytotoxic T cell response, but the big one is the release of these cytokines. And what's really happening here is we have an overreaction. So these memory T cells kind of overreact to this hapten, and they release lots of inflammatory signals, which thereby recruits macrophages, cytotoxic T cells, neutrophils, and all of those cells are going to start releasing inflammatory signals or anaphylatoxins as well. You can see here, this is an example of like a boil from some type of atopic dermatitis. So in type 4 hypersensitivities, there's two primary ones we need to talk about. Of course, contact hypersensitivities, which is that allergic contact dermatitis. Here's where your effector T cells are reacting to haptens. The haptens are bound to proteins on our skin. So particularly in poison ivy, it's called a TCH hapten. That's the toxin released by by poison ivy. And the reason it's so common is because that particular toxin has a super high affinity for proteins in our skin. And so and poison ivy and poison oak release a lot of it. So we get this really intense kind of contact, really high concentration of that TCH molecule. In transplant rejection, we have a couple of different types. Now, this is, of course, going to be some type of organ transplant, and if an organ is transplanted, the less genetically related the recipient is to the donor, the greater the autoimmune reaction is going to be, or the type 4 sensitivity. This takes a little bit of time. It's delayed cell because we have to activate those T cells, right? So that takes a little bit of time. That's why it's 24 to 48 hours, if not a little bit longer. Here, genetic differences in the major histocompatibility complex is going to cause cell-mediated responses to react. This includes cytotoxic T cells or CD8 T cells and your natural killer cells. The greater the difference is, the stronger the rejection is going to be. In allograft, and this is an allograft is when they take a human tissue from one person and they insert it into another person. So that's why it's called an allograft. So they're not genetically identical, but the tissue's been transplanted anyway. So people who receive organ transplants oftentimes have to be on long-term immunosuppressants to suppress the cytotoxic T cell response so that they don't reject the organ that they received. So just a quick review. These are some of the questions that you might want to just try to answer for the hypersensitivity section of this lecture. What are the two stages of hypersensitivity? What are the two different types of type 1 hypersensitivities? Differences between sensitization and desensitization. What's a type 2, a type 3, a type 4? Why is it called delayed cell? Why are firstborn children not susceptible to hemolytic disease of the newborn? How is asthma determined a hypersensitivity? These are some concept questions that I want you to try to answer. You're going to see similar questions like this on your exam. So this concludes our lecture on hypersensitivities. In part 2 of chapter 17, we're going to go into the different types of immune diseases and disorders. Have a great class.